All right, welcome back. Welcome back. We are going to go ahead and continue our Rickoy Talks with Dr. Stephen McCarroll. And after about 10 minutes of talking, we'll have 5 minutes for questions.
Thank you. Thanks for giving me the chance to share this work. This is my RSI class, or at least some of it. We were all reflecting last night over dinner on how one of the best things about that summer was the community we found with a bunch of other nerdy kids. And it was just lovely to have a family like that. This slide is just for the younger people in the room who are at an earlier stage in becoming scientists. It's easy to assume at your age that people who've been successful in a field had a fast and straight and direct path into it, maybe because you've had such a path so far. Reaching milestones early and at a young age, that's not true in my case, or in that of many creative scientists I admire. I kind of meandered my way into biology many years after I graduated from college and had spent the first few years of working in other areas, economics, math. So I just say this, don't worry if you find a mismatch between what you become excited about and what you feel like you've always been good at or expected to be good at or something. Follow your excitement and evolving interests, and you will be deeply rewarded with a sense of calling. And a sense of calling and a sense of impact are perhaps the two best things that work can give you. And don't worry about what you've always been good at. Just follow what you love. So I have a research lab at Harvard and the Broad Institute. We studied the genetics and biology underlying human brain disorders. Over the years, it's been a pleasure to host many rickoids for research projects in my lab. 5 of them are listed here. And 2 of them, pictured here, subsequently came back to the lab and worked for many years as undergraduates and then as PhD students. I'll tell you a quick story about one of them, Nolan Kamitaki. Nolan is the best scientist ever to train in my lab, including me. I admire him so much. As an undergraduate in my lab, he discovered the biological reason underlying why men tend to get disorders like schizophrenia more than women do, and why women tend to get autoimmune disorders like lupus more than men do. He's just an extraordinary scientist, now pursuing his PhD in my lab, and no one has done more to influence the culture or the intellectual vitality in my lab over the years than this rickoid has. He actually, for the last 10 years, he's the first person I go to with a new idea to see whether he thinks it's a good idea or not. You might never have guessed that from his 5-week time in my lab during RSI. Because what I remember about that one summer is that Nolan fell asleep during half of our meetings. But on a good night's sleep, he's perhaps the best scientist I've ever met. And I came to know him through this program. And actually, I know a lot of great biologists and geneticists who, it turns out, are RSI alums. What's funny is that I often knew them for years and even collaborated with them before I realized we were both rickoids or RSI alums. In fact, it was only a few hours ago that I discovered this about William Yang, a terrific neuroscientist I've admired for years, who's been a pioneer in research on human brain disorders and who's giving a talk later this afternoon. So I want to tell you guys about something that we've recently discovered about Huntington's disease, a fatal genetic brain disease. So Huntington's disease is a devastating brain disease. It's entirely genetic in cause. People who inherit the disease-causing genetic variant or allele, they are healthy for many decades, perfectly healthy. And then they develop chorea, the involuntary dance-like movements that patients start to experience at disease onset. But the truly awful symptoms come later. Chorea gives way to paralysis, depression gives way to psychosis, and it progresses over 2 decades and it is always ultimately fatal. In the US, about 30,000 people have Huntington's disease, HD, and a larger number, about 200,000, are at genetic risk to get it later because they have a parent or grandparent who had it. And if one of your parents has it, you have a 50% chance of getting the allele. This is the illness that famously felled Woody Guthrie, the singer, songwriter, and activist who wrote, among many other songs we know and love, "This Land Is Your Land." And as a scientist, there's something really extraordinary about this disease because there's this huge population of people that know it's in their future or know it's in their siblings' future or their children's future. And they come to scientific conferences. And every time it's a reminder of what's what's at stake, because it's rare that you see people while they're normal and healthy and have these healthy relationships and families before they get a disease, and you realize what's going to happen, and you realize that every year that it takes us comes at an extraordinary human cost. And this is true in very many diseases, but Huntington's disease always reminds us as scientists about this. So Huntington's disease involves the death of specific kinds of neurons in the brain. Only, and this is really key, only certain types of neurons are lost, but these neurons are abundant and important. You can see on this brain scan the atrophy that results from their loss. A principal type of neuron that's lost are what are called spiny projection neurons, or SPNs. It was discovered in 1993 that Huntington's is caused by a DNA repeat, the letters CAG, 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 a variable number of times in the gene that was named then the Huntington gene, or HTT. It's variable in length in all of us, and most of us inherit alleles with 10 to 35 CAGs, and inheriting an allele with 36 or more CAGs causes Huntington's, but only decades later after it's inherited. And the long-standing model has been that the inherited disease-causing allele encodes a toxic protein that takes decades to make neurons sick. So the mysteries about this disease have been what's toxic about the alleles patients inherit, why does pathology take decades to start, and why do only some kinds of neurons die? And yet 30 years after the gene was discovered, these questions have been surprisingly hard to answer. So to analyze cell type specific biology, our lab several years ago invented technology for analyzing gene expression, what genes a cell's using and what genes it's not using at single cell resolution. Before that, if you wanted to study that, you had to kind of take a piece of brain tissue and throw it in a blender and study count the molecules in the smoothie. But we developed a way that you could analyze the molecular composition of each individual cell by encapsulating each cell in a tiny, tiny water droplet in an oil aqueous emulsion. These are microfluidic devices that we invented for encapsulating thousands of cells a second in these tiny little water droplets so each one can get its own molecular analysis. And what that meant is we could now analyze brain tissue not as a smoothie, but as a fruit stand, where you could analyze the molecular composition of every individual piece of fruit. In this project, we analyzed, using this technology, gene expression in more than 1 million brain cells from 50 persons with this disease. And we also developed a way to measure the length of the DNA repeat in the individual cells, because earlier work had discovered that it's mosaic in people with Huntington's. Different cells have different repeat lengths. We discovered that the Huntington-causing DNA repeat expands somatically, but in very specific kinds of neurons, which in every patient, there's 4 patients shown here, each one's a column, it expands in the exact same kinds of neurons that are the ones that die in Huntington's. And we think this explains this big mystery of the cell type specific vulnerability. Why did these cells die in Huntington's and not all the other ones? We think it's because these are the ones in which the inherited DNA repeat is getting longer and longer throughout a person's life. But the big surprise that we just discovered a few months ago is that this DNA repeat distorts a neuron's biology, but only after it's become extremely long. 150 repeats, far longer than the length at which it's inherited, and in fact, far longer than the length at which it's usually studied scientifically in many kinds of assays. The DNA repeat takes decades to become this long, and we think this is what explains the late disease onset. This is why people live a healthy life for decades and then get so sick. So there were these mysteries, longstanding mysteries about Huntington's. One of them was, what is toxic about the alleles patients inherited? We think the answer is perhaps nothing at all. And that's why it was such a hard question to answer for so long. Why does pathology take decades to start? We think that's because somatic expansion from 40-ish to 150 CAGs takes decades. Why do only some kinds of neurons die? We think because the DNA repeat expands in exactly these neurons. So our lab now believes that the pathogenesis of Huntington's disease is actually a DNA process for almost all of a neuron's life. So if that's the case, could future medicine slow this ticking DNA clock? So the reason we think this DNA repeats expand, and this is based on work on others, is that they come together imperfectly with the wrong CAG mispairing with the other sequence on the other strand, and that leaves these little bulges. These bulges are recognized by our cell's own DNA maintenance enzymes, and in their effort to fix what looks like a mutation, they actually cause a mutation. They try to repair the bulge by cutting one strand, chewing it back, and using the other strand as a template to fix it, but that causes expansion. So our own enzymes are complicit in this process. But what that means is that you could develop a drug to target those enzymes. And our new goal, and we're interacting with at least 5 companies are now developing medicines that try to prevent Huntington's disease by reducing this enzyme's activity. So finally, I would just like to profoundly thank the Center for Excellence in Education and its supporters, and above all, Mrs. D and her team, who have done so much over the years to create such a wonderful experience for so many kids. Really, in many ways, a life-changing community and set of ideas and experiences. I want to thank the many smart kids that this brings together, smart kids who set their goals high, have adult aspirations, and want to have impact when they're 16, not when they're 25. And finally, the friends and community that have come together around this program. So thank you very much.
Thank you so much, Dr. McCarroll. Now we'll have 5 minutes for any questions that the audience may have.
Yeah.
It takes only one parent to pass the gene on. It doesn't have to be 2.
That's exactly right. That's exactly right. And if one's parent had Huntington's, then they typically have a 50% chance of having gotten that allele or not.
And there is a test for it.
There is a test for it, but today only about 15% of at-risk individuals get it, because since there's nothing you can do to prevent it today, most people decide they would rather not know. About 15% of people decide they would like to have the information. It can be very helpful for family planning, career planning, financial planning. But because today there's nothing you can do, 85% of at-risk people decide not to do the test. Yeah?
Instead of targeting the enzyme that repairs the DNA in humans, would it be possible, do you think, to approach it with like an endonuclease such as CRISPR-Cas9 to kind of get rid of those edits?
It might be. It might be. That's another possibility, yes. Or also to use that kind of tool to knock down expression of that enzyme that's doing that. Yeah. Of course, in some ways, ideal might be a small molecule that someone could just take as a pill every day. But yeah. Yes.
Is it possible for it to become a skip generation?
For it to skip a generation? Not that we know of, although sometimes the age of onset can be variable. So sometimes it can appear to skip a generation because someone may die first of some other cause before they would have gotten Huntington's. But in general, we think of it as fully penetrant. It's just that in some people, so it turns out the longer the allele you inherit, the earlier in life you develop the disorder. But people who inherit ones that's just like 38 or 39 may not actually get the disorder till like their 80s. And by then, someone actually may have passed away of something else. So in those cases, it can kind of appear to skip a generation. But the gene didn't skip a generation. Yeah.
Normal cellular process, and the reason that the disease onsets in people who inherit a longer stretch of CAGs is just
Yeah.
is that what's going on?
Yeah. This is a normal cellular process of DNA maintenance, and most of the time it does a lot of good things. It does this unfortunate thing, but really only in those people who've been unfortunate enough to inherit an allele that's long enough to be unstable in this way. So mostly those enzymes are doing fine. We do think, though, that it's going to be safe to target them therapeutically, because about 1 in 1,000 of us is walking around with a mutation in one of the genes in that complex, and they don't seem to have elevated risk of cancer and things like that. Yeah.
Given that it could take longer for a few people to actually present the symptoms for the PhD to actually manifest, are there any studies done to see if any lifestyle choices would modify those number of years that it's postponed?
It's a really good question. I don't think we know of any lifestyle factors today, but it doesn't mean that they couldn't be found. Yeah.
Dr. McCarroll, you pioneered single-cell sequencing, and now you've got innumerable applications. What do you foresee the future of single-cell sequencing, and will be useful for diagnostics on an individual level and endpoint?
Yeah, so it's a really good question. So the single cell sequencing is actually already being used to make a human cell atlas and to make atlases of the human brain and different organs and things like that. So scientifically, like thousands of labs are using it now. Clinically, it's still the early stages in terms of its adoption. But I think as you brought up, like there's the potential that it might be interesting, or that might add value for diagnostics and allow new kinds of diagnostics and things like that. Applications like that are really at the cutting edge today.
Problems with binary.
To follow up on the last question, our lab has used single cell in order to map out where RNA transcripts are happening, to see which cells COVID is infecting and what it's doing in those specific cell populations.
Great.
Something that's become more affordable for people to think about as a diagnostic test?
It is. It's, it would be nice if it became ever more affordable more and more quickly, but so it is becoming ever more affordable, not as quickly as we'd all like, but yeah. Thank you.
McCarroll.